Thank you to the board at MGMI for inviting me to give this talk again this year. I do sit on that board, but still they're not obligated to ask me to speak, and I do enjoy it when I get to each year. It's really been a pleasure of mine to see this conference grow over the years. Again, I'm Amit Sachdev, and I hold a couple different titles. I work at Michigan State University, and the healthcare arm of Michigan State University is MSU Healthcare. So for MSU Healthcare, I'm one of the chief medical officers. And then for Michigan State University, I'm the medical director for neurology and ophthalmology. I'm the division director for neuromuscular medicine. I'm the clinic director and the grant recipient for the MDA Center. And I'm the center director for our neuromuscular research program. And an absolute honor to hold those titles and to serve the state in this way. And so I'm really, really pleased to give you this talk today. And as you can see, there's my email address and our clinic line. So I do have disclosures. I am a very active clinical trialist, and so we run a number of clinical trials and have run in the past a number of clinical trials. And so you can see all of our industry sponsors that listed there in former and current. I want to emphasize none of those clinical trial contracts are with me personally. They are between the clinical trial sponsor and the university. But my career benefits from that participation, and it's what's expected of me. I do serve on a number of speakers bureaus, helping to educate other physicians around the country. I do receive funding for my clinical and research activities. And I do sit on a number of nonprofit boards, committees, and task forces. And so you can see the list between national organizations, the County Health Department, Michigan Department of Health and Human Services. And then occasionally companies ask me for my opinion to help guide their future development. And so those are what the advisory boards are. And I do sit on a number of advisory boards. And so these are my disclosures where it is. So a lot of this is in myasthenia gravis. So as we get to those areas, I will try to call out where I do have a conflict or a potential conflict. Okay. So the topic today is about clinical trial activity. So we've heard throughout the conference about existing therapies, but this hour is focused on emerging therapies. So myasthenia gravis is really a major focus of development. And what contributes to that is that there are proven outcome measures, so ways for us to measure the disease activity. And those outcome measures are patient-driven. So many of the individuals who are attending today will know about the MGADL, the Activity of Daily Living Assessment. That assessment is really well validated, and it reflects the patient's journey. And the Food and Drug Administration is very interested in measuring the patient's journey when it comes to the effectiveness of medications. And so in myasthenia, we have that. We also have a way to measure the physical exam that's called the QMG, and that is also really well validated. And that takes the physician's perspective of measuring strength and force of breathing and what have you. And so those outcome measures make myasthenia a really studyable disease. But beyond the ability to measure the disease, there are gaps. There are gaps in treatment outcomes, so patients are underserved, and there are gaps in access. I don't think it's a surprising statement to anybody who's attending this meeting that some of our newest myasthenia drugs are amongst the most expensive medications on the planet. And there are reasons for why that is. That myasthenia is a rare disease. A number of these drugs are novel to the treatment of this disease. And so to some extent, there's just a numbers issue here. So some of these are amongst the most expensive treatments in the planet. And because of that, we have sometimes challenges affording therapies. And so gap in access or gap in outcomes are reasons why patients with myasthenia will sometimes consider a clinical trial. So there are many trials. And so for this year, I had to figure out how was I going to tailor the talk. And I thought that I would focus in on clinical trials that are recruiting in the Midwest region. Rationalizing that most of our participants may be from the Midwest. And so my hope was to capture clinical trials that are in the recruiting phase, at least from the publicly available data, that may be of interest to this patient population. So we're going to talk about 8 different ongoing clinical trials. This represents 5 different approaches to how to treat the disease. And amongst those 5 different approaches, there are 6 or 7 different strategies. So really very little overlap between these medications, and a lot of really novel thinking around how do we treat myasthenia. So before I go into the actual clinical trials, any thoughts, comments, questions, things that the audience is really hoping that I will talk about or touch on before we get into these 8 development programs?
I know that there were some questions about the stem cell studies that are ongoing, but I didn't think that any of them were in the Midwest or, this area. There was a question about that though. And anybody who has a specific question, you can unmute yourself now. You have the ability to ask that question or answer that question. If we don't have anybody say that, I'll suggest we go ahead.
Okay. So when we think about stem cells, I did not include any slides about stem cell therapies. When we think about stem cell therapies, we're really thinking about scenarios where we would deplete a patient's bone marrow and then replace it or with a fresh, essentially, transplant. And that strategy probably would work to treat autoimmune diseases, but it's really dramatic. And so the 5th or 6th treatment that we're going to talk about, which is called CAR T cells, is a really interesting way to engineer cells and fight this disease. And those of you who are interested in stem cells, let's focus in on that conversation around CAR T cells, because that might be addressing perhaps the underlying curiosity, but doing so in a much more tailored fashion that's not so dramatic.
Sounds good.
Yeah. Okay, so complement inhibitor strategies. So there is very little question that complement inhibitors help patients with myasthenia gravis. The responder rate across clinical trials is roughly 70%. And our center at Michigan State has been lucky enough to participate in one of these trials that has led to an FDA-approved agent. We were on the ravulizumab study. And there are a number of studies coming down the pipeline to further expand complement inhibitor strategies. And so we're going to talk about 3 trials that are active now in new complement inhibitors. Okay, so this is a busy slide, and I'm not expecting the audience to internalize it, but the gist of this is at the bottom, you have direct damage to whatever organ you're targeting. And along the way, you have some inflammation and immune modulation, and these things feed back on this pathway and drive it forward. So this is a diagram of the complement pathway, and it is blocked at certain locations in order to stop it. And the idea is if you stop this pathway, then you will stop the feedback, right? The damage directly to the organ, the inflammation that builds up, the activation of the immune system. So if you stop the damage, you'll stop the feedback, you'll stop the loop. And again, that strategy does seem to work really well. You'll see this big yellow star right here in the middle of the screen. That's where current FDA-approved therapies for myasthenia act. So this is where eculizumab and ravulizumab, known commonly as Soliris and Ultomiris, that's where they act. And so the new therapies are a mix of either agents that act here or agents that act somewhere else. And again, we're focusing just on clinical trials in the Midwest. There are a number of companies that have clinical trials that are working on different targets on the complement pathway. But this is the first one that I'm going to show you. This is a combination therapy, polizumab and cemdisiran. And this is recruiting at Wayne State University. I do not know the exact recruiting status for all of these studies, but I think this one is still enrolling. You can see that if you're in Ohio or Indiana, there are also 2 sites down there. If you're thinking about, you're in the Metro Detroit area and you're thinking that the Wayne State might be convenient to you, this is the contact number.
Dr.
So,
Sachdev,
yes.
question of whether any of these studies are recruiting seronegative patients.
Yeah, so the ones that I know, I will tell you.
Okay, great, thank you.
I don't know every one of these studies in and out. Some of these studies we are recruiting, and so those ones I know cold, but the ones that I know, I will tell you. Okay, so what do you need to know about this study? So this study is 2 drugs. That's really novel. Now you could ask yourself, well, geez, why do I need 2 medicines to do the same job as one? Right, because these medicines are inhibiting complement. And when you're on eculizumab, also known as Soliris, or ravulizumab, also known as Ultomiris, you're only getting 1 drug. So in here, you've got 2 separate companies, Alnylam and Regeneron. Regeneron makes polizumab and Alnylam makes cemdisiran. And the idea is that the polizumab works very similarly to the way that eculizumab or ravulizumab works. But the cemdisiran, this works totally differently. Polizumab is actually recently, very, very recently FDA approved. So it was approved in August for a different disease altogether. Cemdisiran is not approved anywhere for any indication. So the idea here is 70% of patients, roughly, are going to respond to a complement inhibitor. Can you get that number up higher? And is a combination therapy one potential way to do it? Now, I will tell you, again, I am not recruiting to this particular study, but I'll tell you that most of the time when you're recruiting to a complement inhibitor study, you're not allowed to already be on a complement inhibitor. So if you're on Soliris or Ultomiris now, and you're getting a partial treatment effect, and you're thinking, well, hey, maybe this strategy of a combination strategy might help me, you would probably have to go off of your current medications for about 6 months before they would let you into a study like this. And that just is not the right answer for most patients for their healthcare. So now, if you're not on a complement inhibitor already, and you're wondering to yourself, could a complement inhibitor be the right answer for me? You're always going to try to see if FDA-approved treatment options are the right answer first. And then if your provider is not comfortable with them, or there's a cost barrier, or there's some other barrier, transportation or what have you, then you're going to think about clinical trials. And this is one option. This is a proven treatment class, right? So you'd expect complement inhibitors should work. but both of these drugs would be brand new to myasthenia. And so you're just not sure whether or not they would actually work for myasthenia. Now, this study, this is the Alexion 1720 study. So you'll notice Alexion. Alexion is the manufacturer of Soliris, eculizumab, and Ultomiris, ravulizumab. And this is a complement inhibitor from Alexion, and it is subcutaneously administered. So the Soliris and ravulizumab, or Ultomiris, those are both infusions. This drug is administered into the abdomen, right underneath the skin, as a once-a-week injection. We are recruiting for this study. You can see the red dot right there in the middle. So we are an active recruitment site for this study, and it is actively recruiting. And this one, unfortunately, is not recruiting to seronegative patients. The agent is, so ravulizumab was essentially a re-engineered version of eculizumab. So Ultomiris is a long-acting version of Soliris. I can't say that statement about this drug. It is made by the same company, but it is I would consider it a different drug. And so this is a 24-week study. This is one we're recruiting to. This is, again, a complement inhibitor with Alexion, and it is actively recruiting. Any questions on those two traditional complement inhibitor drugs? So C5 targets.
There was a question for the previous group is, what if you're on a C1 inhibitor?
So, okay, so let's go to this diagram right here, okay? So we're gonna get a little bit into the weeds here. Classic pathway, right? Versus lectin versus alternative. These are the three ways that you can trigger this pathway that leads to damage to the tissue and inflammation and drives the immune system forward, right? So you got to start the pathway somewhere, and here are your three on-ramps. You can see in the classic pathway up here, the immune complexes. That is antibodies. So most autoimmune diseases will trigger here down this C1 branch. And so there are some companies out there that are working on either C1 or C3 complement inhibitors. And the idea is the higher up you target, perhaps you preserve some of this other immune modulation. And so that's why you might think of moving from C5 to C3 or C1. And there's some logic to that. Theoretically, and this is again theoretically, the higher you go up, you may reduce the risk of infections from meningitis or pneumocystis. There are three strains that we think of as encapsulated bacteria: meningitis, pneumococcus, and Haemophilus influenza. And those three strains we think of as more likely to infect a patient and cause a patient harm when they're on a complement inhibitor. Theoretically, the higher up you go, the lower the risk. I would say theoretically because I would still immunize a patient on a complement inhibitor if they were at C1 or C3, or as we'll talk about in a little bit, factor D. So there's a theoretical advantage, but in reality, I don't know if there's going to be an actual advantage.
Okay, thank you.
All right, so
Can I ask, please, what
yeah.
What is the name of the medication with the ALXN study?
Yeah, so this ALXN 1720, that is the study drug designation. It does have a name, but we don't use that name. And so I will have to actually look it up real quick. Here we go. I might have to type this out in the chat. It is Grefulimab. Gefurulimab. Grefulimab. So that's the name of the Alexion 1720 agent, though it's really very early. It's not approved anywhere for any indication, and so we refer to it by its study drug designation. I do have to say, this is the second study that we will have undertaken with Alexion. Studies with Alexion are very well organized, and that's not just like I do have a conflict of interest. I do some consulting for Alexion, but that is earned PR for them. Their studies are very well organized and very thoughtful in terms of recruitment criteria and burden on the patient. And so that has been an impression of mine. So on to the other way that we're studying how to disrupt complement locally here in the Midwest. And the other way is to hit this factor D. So you can see you go down this pathway, right? So the immune complex triggers the pathway, and then it goes down to C2, and then it goes over to C3, and then the C3 splits, and it's supposed to drive the rest of the pathway downwards to C4, C5. Well, this factor D is supposed to help that C3 splitting. And if you block here again where this yellow star is, then you will pause the C3 splitting and you won't go further down the pathway. And so the idea here then is if you block here rather than down here, then you should have the same effect. I don't know that there's actually any other advantage other than have the same effect. I don't recognize a different advantage. So why then would you want to do this? Why would you also want to block factor D instead of C5? And it turns out that actually the approach may not be instead of. This is a diagram from an article in the journal Blood. It's from a different disease, PNH. And for PNH, the complement inhibitors are really the drug class. And so if you don't respond well to this drug class, you may need to boost the effect of the drug. And so this agent, danucopan, is a factor D inhibitor. And what they did in this study is they tried to boost the effect of Soliris, eculizumab, by adding danucopan. So when you look at the pathway, you say to yourself, well, I'm not really buying anything new with blocking here. And in fact, that may not be the treatment approach. The approach may be to package this drug with Soliris, again, to address that 30%, maybe suboptimal responder rate. Those 30% of people who are just not hitting the mark. Now, it's important to think, though, why, right? Why is it that you have people who don't respond to complement inhibitors? And that's because while you are driving down the inflammation and the tissue damage, you're not doing anything to the antibodies that are a part of the disease, right? Then the antibodies are not supposed to be there. So the antibodies are binding to muscle, and they're blocking the muscle, and they're having direct effects above and beyond their ability to kick up inflammation. And so it would be my thought that those 30% of people who are not responding to a complement inhibitor are probably having some contribution to their disease from just having the antibodies around. And that other treatment approaches fix that issue. So my thought here is that adding a factor D inhibitor might buy you some improved response, but may not buy you enough to stick with this drug class. And so one of the drugs is called denucopan. This is also, Alexion owns this drug, and it is their clinical trial agent 2040. And then its cousin, Alexion 2050, is in clinical trial locally. So you can see there's a study site in Milwaukee, a study site in Cincinnati, and a study site in London, Ontario. And so this, again, would be a complement inhibitor. It would be a different approach. It may work on its own, or it may be packaged ultimately with another complement inhibitor to boost the degree of complement inhibition and the durability. This is, I believe, a study that is recruiting. But again, this approach to complement inhibition is not FDA approved for any disease. So this is a new way to block complement. If you're thinking about an existing way to block complement, that's the two previous studies. So focusing on C5 is what the two previous studies do. So study 1720, and then this Nimble study over at Wayne State in 1720 at our site. Okay, so this is a new way to block complement. So we said, all right, that complement inhibition is one way to manage myasthenia, but it leaves the antibodies intact. So the next two drug classes will deplete your antibodies. Now, that is a more robust injury to the immune system, because these are not specific to just the antibodies that drive the myasthenia gravis. So this next drug class, the B cell depleting agents, when you give this drug class, you knock out way up here, right? You don't have immune complexes anymore because you don't make antibodies like you used to. And so you cannot trigger the pathway if you're not making antibodies. The problem, though, is that these antibodies that you make are some of them are useful, right? So you get your flu shot or your COVID shot in order to treat or prevent those infectious illnesses. And because you get those shots, you produce antibodies against those infections. And when you produce antibodies against those infections, if you get infected, you have milder illnesses, or you don't even notice that you got sick with exposures. So we do a lot to train our immune system to make useful antibodies. Similarly, many of the individuals on this call will have had the chickenpox. And then they would have subsequently had dozens of exposures to the chickenpox. All right, most of us would have gotten it when we were 6. Every one of our classmates, our siblings, our cousins, and then when we had children, our children. So all of those exposures, and yet we would have never developed the systemic rash of chickenpox again. And that's because our immune system has been producing antibodies against the chickenpox ever since we were exposed. With a drug like this, you wipe out antibody production. And so it is a clean slate. You lose those immunizations, you lose that memory. So you would say to yourself, well, why would I want to do that? Well, first off, it works. So the drug in front of you, bilzilumab, also trade name Uplenza, is being studied against myasthenia gravis. It is already FDA approved for another neurologic illness, NMOSD, and it works. Patients get better. So if you're in a scenario where your disease has become life or organ threatening, and you need a strategy that is very likely to work. This strategy, generally, we feel does work. And so that's why it's a reasonable bet that it'll probably work for myasthenia. I presented on this last year, and so I think that this trial may be wrapping up at this point, may not be enrolling new patients. But there are other scenarios where beyond myasthenia where you could think of maybe this is a good idea. If you have multiple autoimmune diseases, right? Let's say you have myasthenia and psoriasis, myasthenia and alopecia, myasthenia and lupus, myasthenia and Crohn's disease. So if you have other autoimmune diseases and those other autoimmune diseases are antibody mediated, it might make sense to attempt to deplete production of antibodies across the board and try in one treatment to treat the cluster of diseases. That would be a hope, not a FDA approved phenomenon. But in part because it's not studyable. You just don't have a lot of patients with multiple autoimmune diseases and then ways to measure each of them in a highly reliable fashion. But that's where you would think about this kind of drug. So it's a dramatic step to deplete the ability to make antibodies, and the immune system heals after a while. In my mind, it is probably effective, and it might be the right answer if you have multiple autoimmune diseases. And so again, I think that this trial is probably wrapping up at this point. You can see there are these 4 centers across Ohio and 1 in Pittsburgh, none in Michigan, Indiana, Illinois, or Wisconsin, 1 center up in Toronto. All right, so moving along. Okay, so FcRn inhibitors. All right, so we've heard about this over the course of the meeting. There are now 2 FDA-approved FcRn inhibitors, and these drugs deplete antibodies from your circulation. So you do get to continue to make antibodies, and so that infrastructure remains in place. But the antibodies that attack you get depleted out of your bloodstream. So again, we're focusing up here. This was the complement cascade, right? And we said that the complement cascade had a lot to do with the inflammation and burden of illness and myasthenia. And again, we're hitting it very, very high by reducing those circulating antibodies. And so there's a lot of interest in doing this, and there are 2 FDA-approved therapies now, and several more on the way that are under study. The FcRn inhibitors do appear to work, and from preliminary data from the Vyvgart studies, the seronegative patients may in fact benefit from this strategy, though those signals were not strong enough to get an FDA approval. This study is a study of nicolimab. We were involved in a study of nicolimab. This drug, we were involved in the phase 2. We are not involved in this iteration, which is the phase 3, and that's because we're in the next study that I'm going to talk to you about. The nicolimab study may be ending its recruitment. I took a phone call from a patient in Ohio, roughly right here where this junction of these 2 highways meet. And I was talking to them about recruitment to my study, which is the next one I'll talk to. And I asked them if they had considered going to Ohio State for this study. And we tried to reach out, and Again, I feel like this might be closed at this point. Though if you are in the Ohio area and you are interested in FcRn inhibitors, and for some reason you don't have access, or there's a copay issue or what have you, Medicaid spend down, and you've got some of those challenges and that you're not bridging the gap, I would reach back out and see if this study could meet your needs in order to get you exposed to an agent from this class, and if you would like to participate. So this study is with an FcRn inhibitor from Janssen. Did get through its phase 2 with positive effect, and now it's in the larger phase 3. This is the same agent recruiting in pediatrics. And here I want to highlight my friend and colleague, Dr. Erin Nyburn, over at the University of Michigan. She is the one recruiting to this study in pediatrics. There are so few treatment options in our pediatric population that I do feel like this study may be of use to our pediatric patients. And I want to emphasize that, that this is the location, the University of Michigan. This next study is batoclimab. Batoclimab is a study that we are hosting at our site. This study is a study that does enroll seronegative patients. And so there has been some interest from our seronegative patient population. There are parameters around seronegative patient enrollment. So for my site, we have one seronegative patient enrolled right now. We have to get one more seropositive patient in before we can enroll another seronegative. And so we're on that path. We have a seropositive patient coming in for screening this week on Thursday, and then we should be open for another seronegative. There is another site over in Cleveland at the Cleveland Clinic for this study. So batoclimab is an FcRn inhibitor, and it is subcutaneously administered. So this is administered once a week in the abdomen. And this is an actively enrolling trial. I want to point out that there is a efgartigimod study, that's Vyvgart, that is being hosted in Chicago. I believe this study is closed to enrollment because it's the extension of a previous study. But I want to take a moment to highlight Dr. Koptier's. I think we have amazing talent in the state of Michigan when it comes to pediatric neuromuscular. And pretty much anywhere you live, you're going to have relatively good access. In Grand Rapids, you're going to see Jenna Kruger. In Lansing, you're going to see Cabello Thusang as a part of my group. And in the Detroit area, you're going to see Megan Harper at Beaumont, or Aaron Neal Kneiber at the University of Michigan. So you've got some great talent for pediatric neuromuscular. But if you're looking for a physician that has experience with FcRn inhibitors in pediatrics, it's going to be Dr. Koptier's here in the Chicago area. Because she was the principal investigator on this study for argenx on the Vyvgart study. So I just wanted to highlight that for anyone in the audience who does have a little one with myasthenia and was contemplating this drug class. This might be someone who has direct experience. So our next topic is going to be CAR T cells. All right, so CAR T cells, this is an area that I'm very excited about. We have had some conversations with companies that are developing CAR T cells for myasthenia. And so I have to be a little bit cautious because there are non-disclosure agreements around those conversations. As you can imagine, it's a very sensitive area for drug development. But what I will say is I really like this treatment approach. And if I had to bet on a treatment approach that could knock a patient into remission, this would be it. The CAR T cell strategy is to engineer T cells to attack the B cells that are making antibodies. So an antibody is produced by a B cell, and T cells are killer cells. They go and they directly attack a target. So here you're training T cells to go deplete the cell line that is making the antibodies that's driving the disease. So that's really exciting. And the reason it's really exciting is because you might be able to leave the rest of the immune system intact. So with the previous B cell depleting clinical trial, we talked about how your memory goes away, right? Your training with the flu shots and your COVID shots and your previous exposure to the chicken pox, how that's all trained your immune system. And then if you get a medicine like rituximab or like the clinical trial agent that we discussed, you wipe that slate clean. Potentially with CAR T cells, you don't wipe the slate clean. You just attack and deplete the cell line that's causing the problem. And if you can do that, attack and deplete the cell line that's causing the problem, it might not exist anymore. And that's a really exciting thing. So I'm very excited about this class of development. I'm a little jealous that Dr. Small over in Pittsburgh picked up the phase 2 for this study. And we're going to continue to watch it very, very closely. This is a study that when it hits phase 3, that we are going to try very hard to offer as a site here in Lansing and at Michigan State. And so we're continuing to watch the readout of the data. The treatment effect in the preliminary presentations has been remarkable. And so we're excited to see where this one goes. I think that the conversation around stem cells is too big a conversation to try to re-engineer the whole immune system, when instead with this approach, you might be able to just knock out the problematic B cell lines. So we are on to our last conversation of drugs, and that's the interleukin inhibitors. Okay, so if for those of you who have other autoimmune diseases like lupus or Sjogren's disease, you might be familiar with the interleukin inhibitors or psoriasis. So interleukins are inflammatory infiltrates, right? And so if we go back up to these diagrams, right, we're going to back up just real quick to this one. So chemotaxis inflammation signaling, right? That's what happens as a part of C5a. So what if you left this intact and just blunted the ability to actually produce the inflammation? So you left the rest of it intact, but hit right here in this yellow box. Would that be enough to treat the disease? I can tell you that approach is a bit scary if we were to talk about things like multiple sclerosis, where brain damage is at risk. But when muscles are involved, you might have, especially if you got to the disease early enough, an opportunity to just knock out some of the basic mechanisms that drive inflammation, leave everything else intact, and adequately treat the disease. This is an open question. We don't know whether or not this narrow approach to treatment is actually going to work. If it does, it's probably not for your long-standing patient with significant illness. It's probably for your milder patient. And this is the question that this study right here is going to give us a lot of insight on. So the satralizumab study, what has been, it's also one that I presented on last year. And so I would not be surprised if this one is going to be closing soon or is already closed to recruitment. You can see where some of the study centers are in Chicago, Columbus, and Canton, Ohio. And you can see though that this one in New York is already withdrawn. So typically that means that they've closed to enrollment, and sites that didn't enroll have closed. So, but sites that did enroll are still caring for their patients. So we'll have to see where this direction goes. Myasthenia might surprise us. So I did want to say, you know, with all of these options, how do you choose the right strategy? And I'm going to tell you that I think that while there are nuances to these various treatment approaches, most of the time you're not going to make a decision based on the nuances. You're going to make a decision based on what feels like the right balance of risk for your overall health. Taking a narrow approach seems to work for many patients, but taking a narrow approach of just trying to restrict inflammation might not work for everybody and might not work with your other diseases. And so you might want to deplete the immune system to some extent. And then if so, how much depletion? That's a question that you and your doctor are going to have to figure out. And so the nuances between the drugs, I would focus a little bit less on that. And instead, I would focus on the bigger picture of how significant is your illness today? What's the right way to deliver a drug for you? What will insurance cover? What are the copays like? Those sorts of stuff.
Dr. Sachdev, you are
Yes.
just segueing perfectly into our questions. And I don't know if you can see them in chat or I can read them, because we have several about getting into trials, and I'll read those individually, but I'd like to go back to a question about combining one you're on now with another one. So the question is, would combining the use of a drug that improves the health of the receptor sites, such as Soliris, and combining this with a drug that inhibits the antibody development be more effective together?
So yes. So one of the studies at our site is the Zilucoplan study, also known as the raw pharmaceutical study. Zilucoplan has been approved in Japan and in Europe, I believe, and it's pending approval here in the United States. Zilucoplan is a complement inhibitor, just like Soliris and Ultomiris, and it targets C5, just like Soliris and Ultomiris. But the actual drug is engineered in a different way, where it can be mixed with a therapy like Vyvgart or like roxazolamab, Roxy, which are the two FcRn inhibitors. So this newer therapy would not be depleted by the FcRn inhibitors. And the two strategies could be used together. Unfortunately, you cannot use Soliris and Ultomiris effectively with Vyvgart or Roxy. There are ways you could dose them together. It's just very complicated and it's very hard to actually execute. But when Zilucoplan comes to market, if it comes to market, then you might have the opportunity to actually use both in combination, and there might be cases where that's reasonable.
And so there were questions about getting into certain trials. I might lump them, or I'm going to ask them separately for right now, at least. If you're on Soliris, would you be eligible for FcRn trials like Vyvgart?
So the answer is maybe. So Vyvgart is in study right now, just in pediatrics, is my understanding.
Oh, okay.
Not for adults, because Vyvgart's already approved. But there are two other agents, the nicotinamab and the batoclimab, and nicotinamab over at Ohio State and batoclimab at our site. The challenge, though, with the FcRn inhibitors that are under study is that they will deplete Soliris. And so for batoclimab, for that study, the answer is no. You can't be on Soliris and get into the study, because the drug will reduce the effectiveness of the Soliris.
I got it. And I don't know if you want to explain it just a little bit better, that by reducing the effect of Soliris, if we go back to your pathways, how would you explain it that way? Because
Yeah, well, so the problem is that these are placebo-controlled studies. So
Oh.
for the first half of a study, you might not be getting the new medicine. You might be getting a saltwater solution. And so let's say in the first 6 months, you are on the study and you're getting placebo, and so you're not getting anything. You're not going to notice a change in your health. But then in the back half of the study, you're guaranteed access to the drug. And at that point, you would start the medicine and your Soliris would essentially get wiped out. And then if this wasn't the right drug class for you, you would get worse.
Okay.
So that risk of worsening without necessarily knowing which drug you're on and which phase you're in, that is too high a risk. And that's why this combination is excluded from the studies.
Okay. There's someone who had requested that if you could put the slide back up about Botaglimab and how it works, and then wants to know, can it be combined with sub-Q IG?
So batoclimab is an FcRn inhibitor, just the same as, well, not identical, but it works in a very similar fashion to the other FcRn inhibitors, such as Vyvgart or Roxy. So those are the two FDA-approved ones, and the net result is that you deplete circulating antibodies. Now, the problem with a strategy like subcutaneous IG is that the whole idea is you're giving somebody antibodies. And so this would not be a wise combination approach to take somebody on subcutaneous IG and try to use this class of drugs. So the net result is that if you're on subcutaneous IG, you're not going to end up being a candidate for this drug.
Okay, thank you. And we might have already answered at least part of this question, but it's been fine-tuned to ask. It was about the C1. Can you get into studies if you are already on the C1 inhibitor? And this came in a little bit earlier on, so I think you might have answered that. So it's basically you're on C1s. What are your options? Maybe a different way to ask.
Yeah, so if you're on a C1 inhibitor, you're likely on it for a different disease altogether. And you'd have to take it disease by disease and trial by trial, essentially, to understand what are the inclusion and exclusion criteria for each trial. For someone who's trying to enter a complement inhibitor study, if you're already on a complement inhibitor, it's very unlikely that you're going to be able to get into that study.
Okay.
Because the question would be, again, like why combine the effects of these two drugs? It's a lot like trying to take two blood pressure medicines when you could have just taken one to do the same job. For the other studies on this list, it is possible if you're on a C1 inhibitor that you might qualify, but you're really going to have to take it trial by trial.
Okay, this has been just incredible cutting edge information. Thank you. And I'm going to turn it back to Susan. She might have a few more comments or questions, but thank you, Dr. Sachdev.
There was one last question is, can I get into batoclimab study, which I said that wrong, but you can say it right.
Yeah.
If she gets off IVIG.
So yes, but it has to be the right decision for your overall health. And that's where having your physician guide you as to how to manage your myasthenia. This has been a common question that we've received amongst the seronegative patients in particular, because the seronegative patients don't have many treatment options. And so a lot of them are on chronic IVIG. So yes, you could potentially enroll in this study. It has to be a gap between your last IVIG dose and your date of screening into the study. And if you're really a brittle myasthenic, that just might not be the right answer for your health.
Okay, so one question, my doctor is taking me off IVIG. What's the gap? I think you might have mentioned that also after she typed it. How long would I have to stay off, I think is what.
So again, it depends on the study protocols. I don't recall off the top of my head with batoclimab the exact number, but if I had to take a guess at what it was, I think it was 60 days. But this is the sort of question that Aubrey Alexander, whose name is on the screen right now, she would be able to field that and would reply, within hours on a weekday. We don't make her work on the weekends.
Right, and also, Dr. Sachdev, as you may have mentioned before, you could go off the IVIG, but then not get accepted into the study, and then you're not on any treatment, right? So that's
Right. And the other thing is to think, if you go off IVIG and you get into a study and you're in the placebo arm, and the placebo arm lasts 6 months, right? So now so let's say it was 60 days that you had to be off in order to even get into the screening period. The screening period to confirm you're otherwise eligible lasts 30 days. When you're in the randomized portion, I don't know what you're on and you don't know what you're on. And that situation lasts 6 months before you're guaranteed access to the drug.
yeah, okay,
Now you're adding up to 9 months.
right.
So you really cannot be a brittle myasthenic, or
Okay.
it cannot be a area where you rely upon this as your treatment strategy. Otherwise, you're going to be hospitalized. That's just
Yes.
not the right answer for certain people's healthcare.
Yes, thank you for making it that clear. And you have a comment that thanks you because it was the best understandable presentation. Susan, I'm going to turn it back to you because there's a second part to that question that you can handle now, or to decide how you would like to.
Yeah, the question is there a way to keep in contact? If they call Aubrey or follow up with Aubrey, is that something that if they could be added later when it's an open, they could get on that trial?
Yeah, so I would say email Aubrey. So both of the trials that we have right now are actively enrolling, and then we have another 3 trials in the pipeline right behind these 2. We're a very active clinical trial center, and so sometimes it's about timing. And then we do a fair bit of advocacy, right? Being on those advisory boards makes a difference. And we are trying, especially for our seronegative population, to get seronegative arms onto these studies. And so especially if you're seronegative, reach out to Aubrey and we will do what we can.
Thank you, Dr. Sachdev. That makes so much sense because I think that there is that need, and a lot of times they're looking for people seronegative to be included in order to answer those questions, because it is part of the FDA approval process when they approve those drugs. I had one other question about the phase 3 of one of the drugs that you were trying to be on. Which one was that?
So the CAR T one.
Yeah.
So when this trial hits phase 3, we will try very hard to make sure we are a site.
Okay. Does the number of, so one of the things that I think I've heard from other research sites is that knowing people who are possible who want to be in research and have raised their hands and said, and they're pre-qualified by Aubrey or at least that information, is that something that those people might be able to be reached out for other studies?
Yeah, of course. So we maintain a list of patients, and we maintain our recruitment statistics, and that's always very useful when we're talking to study sponsors. Now, if I am doing my job correctly, that when push comes to shove, a patient who is eligible for a clinical trial will end up consenting into it probably no more than 1 out of 4 times. So that means 75% of patients who think they might want to enroll in a clinical trial, when given all of the information about available treatment options and the
Risks.
risks of the trial, will end up saying, you know, let's wait this one out. And so sometimes it's just about sitting down with a provider and having some faith that our center or the center you're being cared for can actually move your care forward. And that's what it takes.
Yeah, I think that's a good point. Figuring out, again, based on your care provider now, is there a way that they can move your care forward with the options available?
Right. Right.
That are,
FDA approved therapies are the right answer for the vast majority of patients.
right. I have to say, you have the patience of Job with what was going on next to you. So I applaud you in that. I somewhat have the same thing when I have not locked my door. And one of them's a furry animal, as well as, you know, as humans. So thank you very much for your persistence in providing that information.